Good afternoon, everybody. I would like to start by thanking PacBio for giving me the opportunity to be here and speak about novel workflows that have been developed in the past year that I believe can couple nicely with the PacBio science. I have to say I was really impressed to see all the advances that are going on, and that's just really beautiful. So the title of my presentation is Rethink Your Cell Lysis with Adaptive Focus Acoustic. So the agenda is divided in several categories. In the first part, I would like to talk a little bit about the AFA technology overview and present the scalable solution that we worked on in the past year. Then we will jump into cell lysis solution, where I will show the data that we collected about DNA, high molecular weight DNA, and RNA extraction, intact RNA extraction. Then a few slides about a collaboration we have with Circulomics for DNA, high molecular weight DNA extraction from tissue. And then a few key takeaways and questions. So adaptive focused acoustics use a sonicator to create ultrasonicator transducer to create sonic energy that is focused. This means that the pressure field that is induced by the sonic waves is focused at the sample size, avoiding loss of energy and optimizing the contact. What this does, under the right circumstances, when you have nucleation size, the pressure field that is represented here that oscillates between negative and positive pressure generates a bubble on the surface. This is a nucleation site, and the bubble grows and collapses, as you can see from this graph here. On the y-axis, there is the radius of the bubble, and on the x-axis, there is the time. So the bubble grows and collapses until it reaches the maximum size, and then it goes through a rapid quenching that generates shear forces. Shear forces are like molecular scalpel. Depending on the power that we use to deliver the sonic energy at the sample, this can be very kind and can massage your molecule, or can be very strong and they can cut the molecule. That is the technology that is behind DNA shearing, that there is now behind cell lysis, rupture in the wall, but also the paraffinization from FFPE sample or dislodging proteins and nucleic acid molecules. So the ultrasonic that is used is the same one that is used also in diagnostic imaging. We operate in this regime for several reasons. One is that the focal zone, it becomes very tiny and can couple well with the vessel. It is very important when using sonication to have not only an adaptive control of the sonication per se, but also to have the right consumable and, of course, the right chemistry. If you have these three components, then there is no limit of what you can extract from your molecule and your tissue. This is an example also of what's going on when you couple sonic waves to a plastic sample, for example. This is a simulation that shows how heat is generated very close by to the transducer that is not regulated, and then is transferred to the vessel by leaving very little or no control on the temperature inside the vessel. This is what happens when you use a probe sonicator that is almost like sticking a very hot rod inside your sample and generating a lot of heat. you do have cavitation, but the point being is that it's very difficult to keep the control of your temperature under these circumstances. That becomes a very, very important component when you think about sample extraction. With the Covaris AFA technology, specifically using a glass tube, the cavitation occurs, but it's very easy to maintain the temperature. And this is done, as I explained before, by using different powers, by using a pulsing mode where you alternate pulses of sonic energy with delay times. So I decided to start after this with this one because this is very known among the PacBio user. This doesn't use AFA. This is kind of the present situation of what Covaris and PacBio are now. And with the Gtube, we can share DNA, high molecular weight DNA, in very producing versatile lengths between 6 to 20 kb, up to 30 microgram of DNA, and gives very reproducible and repeatable DNA shearing, as you can see from the electropherogram of the bioanalyzer or also from the gel. So, but what is the future that Covaris envisioned for the high molecular reads? So the sample preparation is always a bottleneck, and it's very interesting to hear among the audience several questions about how is your DNA reading correlates to your sample preparation. And the answer is, yeah, it's very important that you have a sample preparation that is standardized and allows you to extract very high quality DNA or RNA or even proteins that you know that they are consistent and they represent very well your sample. So if you look at the conventional Lysis methodology for cells, we have bead beating, vortexing, enzymatic, or using chemicals. Then you have an extraction that is through phase separation. You have, again, vortexing or pipette mixing, volume transfer, and then a purification, filter-based, using vacuum, using membranes, pipette mixing, or volume transfer. The workflow that we have been working on, they were focused on being scalable, being automatable, and standardize the extraction. So the lysis and extraction are actually, if we think about coupling the AFA instrument consumable chemistry with liquid handlers, for example, they are done in the same step. We have a fast lysis, it's really fast for bacterial and mammalian cells. We don't use harsh chemicals. We have our proprietary buffer that, depending on what you want to extract, allow you to go in all the downstream applications that are the most common one, qPCR, sequencing, or MALDI-TOF, or LCMS. Then there's an extraction that, like I mentioned before, is automatable and can be directly done with library preparation. For example, if you think about NGS, when we extract high molecular DNA using the ThroughPop buffer, can go directly into the Nextera library prep, and only applying one round of purification that is a magnetic bead-based method, or whatever is depending on downstream application. If you're looking to do qPCR, for example, and you can dilute the sample, you can go directly and perform your analysis. So these are the solutions that allow these tricky points that I mentioned before. We have instruments. We have several instruments. These instruments here, the four actually, they were built for high throughput. 3 of them are good for automation. This one is the latest member of the family, is the R230, that can fit directly into a liquid handler. The LE220R Plus and the RSC, they have a light curtain. This is for high power application or even to treat 384 sample at the time. And they can be coupled with a liquid handler, but you need a robotic arm. While the R230, it doesn't. It can just fit there, and you just need a gripper to interface with that. Fully interfaceable with several liquid handlers. We have drivers for that. There is water management control. So it's basically plug and play. You walk away, you do the lysis, and at the end of the day, you have the library prep if you want to do NGS or long-read sequencing. We have the consumable. This one, the AFA2 TPX, is like a PCR plate. It's a different polymer that is what I mentioned at the very beginning of the talk. It's very important to have the vessel that is designed the right way with the right material that can couple well with the sonic energy. And then there is the glass consumable, the microtube, that is also automatable, even if it is not as straightforward as the AFA tube. And then we have several buffer for several applications that I will describe later on during the talk. So this is also for high molecular weight DNA extraction. This is not high throughput, but it allows to extract high molecular weight DNA from tissue. The way that it works is very simple. You put the tissue in these bags, you flash freeze them, you smash them, and then you collect them into the tube, and then you can either extract your DNA, or you can use AFA to homogenize even further the sample that has been impacted, and extract your DNA and RNA. This is an example of automation. This is an LE220R Plus that is coupled to a Lynx through a robotic arm and a gripper. Specifically, this was a 24 format microtube 130 that, as you can see, it is taken with the gripper into the liquid handler. It goes into the AFA instruments, it gets processed. And this has several advantages because AFA is not only about shearing. What we can do is also mixing, and it looks like a silly thing, but if you can mix using the AFA, you save a lot of space on the deck with pipette tips. So you can go back and forth, and if you think about bit purification, where the more suspension you do, the better your DNA gets, you can do this several times, especially when you want to wash with ethanol. This is automatable with the microtube and AFA tube, as I mentioned. There is water management, autofilling, automatic adjustment, software, temperature control, the gas, and there is also an RFID sample tracker if you are thinking about tracking down your sample. So specifically about the cell lysis, the data that I'm going to show you, it will show lysis and extraction of high molecular nucleic acids of mammalian cells, yeast, and bacteria without using harsh chemical, no bead beating, great temperature control. Bead beating can heat up pretty much, and if you want to lyse tough cells like yeast, then after the bead beater is done, you have to leave the plate there a little bit if you don't want to get burned. There is it is a non-contact extraction. This is great, especially if you're worried about cross-contamination. We have high yield. We have multiple steps that are combined in a single consumable and instrument, and is automatable, friendly, and especially is scalable. So this is the workflow. It's very easy. We start with a sample. We prepare the sample, add in the lysis buffer. If it is from colony, you just pipette the sample in, spin it down, remove the supernatant, put the buffer. If it is frozen, that is actually great for RNA extraction. You just go directly into the AFA by preparing the sample, of course, before freezing it with the buffer. Then lysing with AFA, purifying by extracting the biomolecule with AFA, and then going into your downstream application. So this is an example of the high molecular weight DNA that was extracted from yeast, Cerevisia lipolytica, gram-negative E. coli, and gram-positive Listeria monocytogenes and Staphylococcus epidermis, and also with high molecular human genomic DNA from blood. So this workflow was done literally just by blood, adding the buffer on the liquid handler, do some run of AFA, bit purification, and we get DNA that is a molecular weight that is about 40,000. So the cell count is a standard cell count that customer in the field use. 3 × 10⁷, 4 × 10⁸. For yeast, we went up to 1 billion cells, and the yield were actually pretty good. So for yeast, they were between 50 to 85%. For coli and mammalian cells, they were between 75 to 100%, just based on the cell count. The treatment time depends on the kind of cell. So gram-negative and gram-positive, they are very easy to lyse. We can do 96 or 384 in less than 3 minutes. In 3 minutes, they are completely lysed and they are ready to go into library prep or purification. One of the big advantages of AFA is that it works well with low volume. So even if there is this amount of cell, working with 30 to 50 microliter is actually giving you the best results. So in this is a great way to improve the process and also saving money on the reagent. This particular data was done on the Lead-220 Plus and also on the Mid-220, but like I mentioned before, the settings are easily transferable across all the instruments because the technology is the same. So this is a comparison between Covaris and other solutions that are out there in the field. The preparation time for bacteria is way faster compared to everything else. The lysis volume is 10 times lower. It can be more than that without exceeding 100 microliter, 50 to 100 microliter. It doesn't give you any advantages though, that's the secret. This is automation. As I mentioned already a couple of times, you can think of going there with the sample, with the plate, doing the lysis, program the liquid handler, and have the library prep ready, made right away after purification, reparation, and ligation, and so on. So the cell count, these are what we validated, even if we didn't really see plateauing, so there is still space to go more than that. And for bacteria, is 4 × 10⁸, and for yeast is 5 × 10⁷. So times are excellent for yeast. Actually, now we have like a better way of a faster way to extract DNA from 60 minutes. Now is 20 minutes for a 96 or a 384 well plate. The time is not doesn't scale with that. The processing time is actually the same. For Cerevisia, it's about 15 minutes, and for cells, it's a few minutes. So this is the workflow from blood, and this is also this was done also on a liquid handler by using the TruePop buffer. This is a super simple, simplified workflow. There are 6 reagents that are required. This is in line with other kind of kits that are out there. The reagent volume are changing between 6 to 20 milliliter in total for 96 samples. The reagent reservoir is only one, getting rid of reservoir for more space on the liquid handler. The mixing step is done by AFA without having a vortexer or more pipette inside the liquid handler. There is no centrifugation, and centrifugation it's always the bottleneck for automation. Magnet and with an incubation step. So we have several technical notes that if you are in the booth, and if you are interested, please come and talk to me. I would love to hear more about your workflow and your needs. And this is for this is about the blood workflow, where not only we were able to extract the high molecular weight, but we also did shearing for WGS or for targeted panel. This is for NGS, and so on. So just few words about RNA. We also have workflow about RNA. We did experiment with and without AFA, and without AFA, even if the cell still lies a little bit, the ring score are very low. These are just two different buffers that we use for the purification, showing identical result, super high yield, and with the DNAs, we get rid of the DNA that is in the background. This one, this very nice peak here, is done with the full protocol. So the nice thing of this is of this extraction is that we the RNA in this particular that I just showed you was amplified using the Lexogen teloprime cDNA amplification kit that amplify only DNA that is messenger RNA that is intact, that is capped on one side and there's poly A tail. So after that we did this, we ran the amplified cDNA on top of the human reference DNA, RNA, just bought it commercially, and they overlap almost perfectly. This is suggesting that the extraction of RNA is extremely good, and is really giving lot of information about intact RNA that we believe that this can really be a winner in terms of RNA-seq. So I'm gonna skip this. We have solution also for proteins, as I mentioned before. I just would like to acknowledge, I don't want to go too much into detail, this is collaboration with Circulomics, where we use the tissue, the cryoprep and the tissue tube, and we had excellent result also here with very good N50 and very good throughput with very long read. So the key takeaway is that we can enable all the omics from Lysis to purify biomolecules that are automation friendly with cost saving and maximize the result with minimal sample loss and improve the turnaround time. We are really looking forward to collaborate with you guys with high molecular weight DNA and RNA, because we believe that this technique is superior to everything that is out there and can standardize your workflow. I would like to acknowledge and thanks my colleagues and the people of Circulomics that we work with, and thank you for your attention.